So I actually decided to change the lecture. It will still be covering what was in the title I gave, but a little bit overall writing theme is systems biology of metabolism. But as you will see in the lecture, that covers our studies in several different areas of biology. So first, very briefly about metabolism. It's the core of cellular functions because it provides the building block for building cells. It provides the free energy for cells to maintain functions, but also to expand and grow. As well as it ensures proper flow of electrons. These are primarily handled through cofactors like NADH and NADPH. And so we are interested to study and have knowledge about metabolism for various reasons. We can use that to engineer cell factories. We can use it for diagnose disease and predict its development. And we can also improve energy uptake from food or feed for that matter. And I'm going to touch briefly on all these aspects here with emphasis on yeast metabolism, primarily because it will illustrate how we can embark in combining mathematical models to begin to describe a living cell. So very briefly about metabolism and introduction to this. So we're all familiar with the metabolism of muscle cells where glucose is used as an energy source, can be respired completely, but if you get oxygen limitations, you may have lactic acid accumulation as overflow metabolism. And that's very similar to the metabolism that yeast cells are using when they are making alcohol. They're converting glucose to ethanol. Yeast also has the ability to respire completely glucose to CO2, but it doesn't do that in excess glucose conditions. There it prefers to send most of the glucose to ethanol. And understanding the overall regulation of how the metabolism is split between these two is quite interesting because, as I'll come back to, much more energy is of course harvested when you oxidize the pyruvate completely to CO2 compared with when you produce lactic acid or ethanol. The lactic acid production in human cells is also associated with the Babu effect in cancer cells, which is also quite interesting why they shift actually when they start to grow faster to this type of metabolism. So how do we study metabolism? If we look at a yeast cell, it has about 1,500 different metabolic reactions as we know it today. These are associated with about 900 genes encoding enzymes that are catalyzing these reactions. And if we look at the similar number for human cells, we have about 8,000 metabolic reactions, and these are associated with about 3,000 genes. The 3,000 genes is about 15% of total genes in the human genome. The same for the 900 in yeast. So it's kind of interesting that about 15% of our genome seems to be allocated to this core function in any given cell. So I'll be talking very briefly first to the concept of metabolic engineering, where we are aiming to engineer cells and their metabolism. I'll give a brief example about secondary metabolite production, particularly because it's related to comparative genomics. Then I'll talk about quantitative omics analysis, how we can advance that field, and use that to model yeast metabolism in much more detail. I'll talk briefly about adaptive laboratory evolution because it's an example about how we can use systems biology to learn something for cells that have different phenotypes. And then I'll move on to talk about cancer, and then in the end talk very briefly about modeling of the human gut microbiome. So first about metabolic engineering. So this is relevant in the context of developing a bioeconomy where we are producing fuels and chemicals in a more sustainable fashion from biomass. And in this process here, biomass is degraded into sugars and then they're converted further on in a fermentation process where we use cells as a factory in this conversion. And so there's much interest in this field. The chemical industry and also the fuel industry is, of course, looking into whether they can become independent of oil and use this kind of technology. So how do you do this? Well, you start by identifying a molecule that you're interested to produce. You identify whether there is somewhere in biology an organism that can produce this chemical. then you can either use that natural producer and kind of improve the titer rate and yield, the performance of this. So this is very well exemplified by penicillin production. We found an interesting molecule, and the performance of the original producer of penicillin was improved through random mutagenesis and screening. But today, one often uses platform cell factories where the pathway is reconstructed, and then you still improve the titer rate and yield. There are some success stories. Two of them are mentioned here. Maybe more important here to focus a little bit on the challenges, because it may sound quite easy to improve the titer rate and yield. But if you look at a simple molecule like 1,3-propandiol, it took DuPont Genencor about 15 years and costed them about $130 million just to develop the cell factory. And another example is artemisinin. It took UC Berkeley Amaris about 10 years, maybe $50 million to actually get to a level where you could produce this commercially in a cost-competitive fashion. So why is this so difficult to engineer a cell factory? Well, this is because, of course, metabolism have evolved in these cells to serve the needs of the cell. So yeast cell to produce ethanol very efficiently, and we want to recruit this metabolism to overproduce another compound. We're up against these millions of years of regulation and evolution, and so that is the challenges. So therefore, when we do this, we do it in an iterative fashion. We introduce a design. We then reconstruct that design in the cell. We test it, and we acquire knowledge about it. And so I often say that we learn a lot about biology also in this process here. So even though the objective is very clearly to obtain a cell factory that can overproduce this chemical, but we obtain a lot of knowledge about how the cell metabolism is actually hardwired in that cell, and that we need to understand in order to unlock that regulation in order to overproduce it. So let's look a little bit on an example of interesting molecules to produce. So look a little bit about identification of target molecules. So there is much interest in finding new secondary metabolites, particularly to be used in antibiotics. Antibiotic resistance is a major global problem. Many of the similar chemical scaffolds are identified again and again, so often they don't really resolve so many of the resistance problems. So we therefore undertook a project in going into a group of organisms that have actually not been exploited that much, namely filamentous fungi, and more specifically on penicillia. Even though penicillin was discovered in one of these organisms, very little has been looked into finding other bioactives in this group of organisms. So we decided to take a genomics approach to this. We used whole genome sequencing, annotated the genomes. We then used an algorithm, a computer algorithm called anti-smash to find secondary metabolic metabolite gene clusters. We were then looking into for consensus pathways and see if we could identify also groups of molecules that are produced newly in these organisms here. We combined our sequences on 10 new genomes with 14 other sequence genomes, so we had a total of 24. They covered several different clades of penicillium. There is about 200 different penicillium species reported to date, so it's an extreme diverse family of organisms. So, but here we covered several of the key clades in that class, in that family. And so the interesting thing is that when we look into metabolism and look into the overall annotation, what we see actually that where the number of proteins in different categories, actually these fungi are really specialized to produce secondary metabolites. This is the class with the most number of genes or proteins associated with. It even comes before carbohydrate transport and metabolism, which is second here. And this would be very different from if we look into many other organisms where metabolism of central carbon metabolism will come as a top score. If we look into the total biosynthetic gene clusters in each organism, you can see that approaches something in the average of 55 different secondary metabolite clusters in each organism. And these are spread into polyketide synthase clusters with an average of about 18, and then come non-ribosomal peptide synthases, and then comes various other clusters of ability to produce biosynthesis. So how diverse is the capability across species? Well, so what we did was we did a cluster analysis, so looked into common domains between the species. And you would, of course, find that when we compare one species against the same species, we find perfect match. But you can see we do find these clusters where a certain set of secondary metabolites are actually conserved between the species. And so that means that there are several sets of metabolites that are produced in several species. But actually, there is a very large diversity both in domains related to polyketide synthesis or non-ribosomal peptide synthesis. We could then look into some of these clusters here and identify also are all the genes in these clusters here, are they similar? And because it often involves not only production of the aglycone but also further decoration. And when we did that, we could find that, for example, for certain groups, in this case here patholine, which is a scaffold for antibiotics, we could find a number of different additional enzymes in the clusters to varying, and particularly in this group here, we find that they are missing several genes that may actually result in a completely different chemical structure. We look further into the variance of the patholine cluster. Also, this is interesting how that the sequence, the whole cluster likely did not transfer horizontally back in time because the organization is variant between the clusters. And interesting enough, also we identified a completely new gene cluster in penicillin that seems to be produce ionotones, which is an antibiotic that has traditionally only been found in Aspergillus. But interesting enough, we find a new enzyme here in this cluster here, and we are currently looking into what kind of chemistry that may be associated with producing. So we're also looking into the metabolic clustering with a phylogenetic analysis. So we developed complete models, metabolic models for each of these species here. And you can see that the phylogenetic clustering, the traditional clustering and grouping of these different species based on morphology does not actually match completely with how the metabolic capabilities of these. So this can also lead to that one has to be careful on drawing conclusions on traditional phylogenetic assignment of species to metabolic diversity. So let me then move on and talk a little bit about quantitative omics analysis. So this is very much related to the test analysis. And I'm going to illustrate that from a study we recently did on Saccharomyces cerevisiae, baker's yeast. It's also used for ethanol production, large-scale bioethanol production, but also potable alcohol in various alcoholic drinks. And besides that wide use in industry, it is also a key model organism for studying eukaryotic cells. And what we wanted to do was to see how far can we push the boundary of actually getting quantitative measurements on a cell basis for as many molecules as possible, and can we begin to sum up the total inventory of this cell. And so in this study here, what we did was that we looked into yeast grown at a reference condition, and then we exposed it to 3 different types of stress: heat stress, osmotic stress, and then also stress by having increased levels of ethanol. And in each type of stress, we looked into 3 different levels of this stress. So that gave us a total of 10 different conditions. And we then quantitative analyzed the number of proteins. We did it quantitative, so we have protein copies per cell, as well as messenger RNAs. And we also looked into protein degradation, as I'll talk about. This kind of study gives about 300,000 data points, so there's a lot of data to analyze and structure. But let's first look a little bit on the messenger RNA protein levels, because as I said, we have quantitative data, so we can look at the abundance here in molecules per cell. Here we can see that the messenger RNA is varying here. This is a logarithmic axis here between about 1 copy to about 100 copies. So that's about 2 orders of magnitude, whereas the protein abundance you can see spans from maybe 100 copies up to about 1 million copies. So it spans several orders of magnitude. And this is a typical finding in biology, that you find a larger variance in protein copies compared with messenger RNA copies. For that reason also, if you look at a general correlation between messenger RNA copies and protein copies in the cell, there is a very poor correlation. So this has been found in several studies before also. And this is of course because each gene or each messenger RNA may be translated differently for each type of protein. So we therefore looked into for each gene and took advantage of the fact that we are studying 10 different conditions where there's quite a large variation in expression of each of the genes. And now we can see that when we look at it on a gene point of view for each gene and look at the copies of protein versus messenger RNA, we can begin to find very good correlation between protein and messenger RNA abundance. And here's shown illustrative for just 4 genes. But when we did this across this panel of about 800 proteins that we measured quantitatively, we could find very good correlation. And here's shown the distribution of the Pearson correlation, with the average Pearson correlation being about 0.88, indicating that we have this very strong linear correlation between protein and messenger RNA abundance for each individual gene. That led us to look into what is driving this large variation in translation rate that this will basically translate to. And in order to do that, we first had to quantify degradation rate also. So we did that for all the proteins. And you can see these cells were grown at a specific growth rate of 0.1. And you can see that the approximately average protein degradation rate has about half the rate of that. So that means that about 33% of the protein is turned over constantly. But you can see that some of the proteins have a very, very high turnover rate. Many of these proteins are associated with cell cycle, for example, that really they need to be degraded continuously as soon as they have been used in a certain part of the cell cycle. But overall, when we had these data together with the other data, we could now calculate the protein translation rate for each of the genes that have varying expression at the conditions we're looking at. And then we look for what is driving those translation rates. And it turns out that the key dominant factor is really messenger RNA abundance. So the more higher messenger RNA abundance, the higher translation rate you have. You have a relatively small impact on codon usage, here shown as codon adaptation index, or the translation adaptation index. There is some impacts on specific genes, relatively small from the UTR regions, but we have still an about 19 unknown factor here. But still really messenger RNA abundance is the key driver of this. Interesting enough, this translation kinetic constant is quite constant across the different conditions. We could see that by looking into the protein messenger RNA ratios at one growth condition here versus another condition here. So this is reference versus high temperature. And you can see that besides a few proteins, it's falling significantly outside the diagonal here, that there is a fixed translation rate across these 2 different conditions. And that goes for all the 10 conditions we looked at. This is actually the most largest deviation from that. So one thing that is interesting to remark here also, that we now measured quantitatively all the proteins, but one thing that we clearly find is that the total protein content in the cell is not changing between these conditions here. That is actually constrained, and that goes in general, even for yeast grown at many different conditions, as well as for a human cell, that the amount of protein that we have on a mass fraction basis is really kept quite constant. This is quite remarkable because that means that if we are overexpressing a certain protein, or we need to change the protein composition in order to do other functions in the cell, it means that it needs to be taken from other proteins, so to speak. And so here's a distribution, overall distribution of the proteome allocation between the reference and the high temperature conditions. And you can see that there is some shift. You can see here that what happens when it shifts over to the higher temperature, yeast starts to actually ferment. It starts to produce ethanol. Here it's completely respiratory because of the growth condition we did. You can see that a little bit more proteome is allocated to glycolysis, less to amino acid metabolism. Ribosome is about the same. Chaperones and folding is about the same, a little bit less to oxidative phosphorylation. But if we zoom in on individual proteins, we can begin to see a major remodeling actually of the proteome. And I'm not going to go into details here, but you can see that several glycolytic enzymes are beginning to change their function because the cell now needs to adjust the proteome allocation so it matches actually those metabolic requirements the cell has under these stress conditions compared with the reference conditions. So these kind of data here, we are now beginning to use for modeling, and let me, when I move on here, tell you a little bit about how we can begin to integrate these kind of really quantitative omics data into modeling. So let me move on and describe how we do that in terms of yeast metabolism. So first of all, before I go into the framework for modeling, let's return a little bit to the about 1,500 reactions in metabolism that I referred to in yeast. So we may think of these, and we are taught about these in biochemistry, as individual segmented pathways, glycolysis, amino acid biosynthesis, TCA cycle, and so on. But in fact, the cell doesn't operate like that. Metabolism is highly interconnected. And this is illustrated here, where we have plotted all the different enzymes, as well as the different metabolites that these enzymes are catalyzing the interconversion of. But because many of these metabolites are used by many different enzymes, suddenly two enzymes that may be very far away from each other in metabolism are getting highly connected. And this means, of course, that in order for keeping homeostasis of metabolism, the cell will have to evolve a global regulation on these different pathways here. But it also imposes actually some constraints, because the cell needs to balance the flux between these different pathways here, and this is what we can take advantage of in modeling. So what we do for that is that we use these so-called genome-scale metabolic models. What these models are is it's a comprehensive representation of the stoichiometry of the individual reactions that are occurring inside the cell. We link the description or the catalysis of each of these reactions to the specific enzymes that carry these reactions out. These are the corresponding proteins. It can be protein complexes. We can also have isoenzymes, and that links to genes. So this gives a gene-protein reaction connectivity in these models here. Comprehensively, that results in a network, and we can also have a mathematical representation of this in terms of a stoichiometric matrix. So these models we can use for various uses. We can use them for simulation, because there will be a balancing of the metabolites, each of the metabolite inside the cell. So the fluxes into that pool will equal the fluxes out of that pool. We can use it also because it represents a highly annotated graph of connectivities inside the cell. So we can overlay, for example, RNA data for two different conditions, and then find out how is the metabolic reprogrammed, metabolism is reprogrammed from these two conditions. But we can also reconstruct models for different cell types. It can be a healthy cell, it can be a cancer cell, and then we can see what is loss of function and what is gain of function in these two different metabolic networks. So what we are now working on, and this concept of genome-scale metabolic models, as I said, have been used extensively for many different bacteria, different fungi, but also different type of higher organisms. But what is particularly fascinating and interesting is that we can now begin to expand the scope of these models, because traditionally they only contain information about the stoichiometry. But we are now interested to begin to introduce information about protein level, but also kinetic information, particularly KCat. Eventually, if we have correlations between messenger RNA levels and protein levels, we can begin to incorporate messenger RNA levels. We are also looking into transcriptional regulation, so we can eventually begin to have a comprehensive description actually of how a cell is functioning. So briefly a little bit about the interest of looking into the KCats is that you can imagine that if you have a low efficient pathway here with these KCat values specified here, in order to carry out a flux of 100, you need to allocate, let's say, a sum of enzymes here with 250 here. Let's say you have a pathway with twice the efficiency of each KCat reaction here. You can actually carry out this flux, the same flux, with half the amount of protein. And as protein comes at a cost inside the cell, that means that you can actually really favor pathways that have high and efficient KCat. And that's basically the principle we're using in terms of when we're incorporating KCat information. So we wanted to expand the concept of flux balance analysis to include cost of building the enzymes. for each individual enzyme, because it will also cost more to build a large enzyme, and then also impose kinetic constraints for each of these enzymes. So basically there becomes a penalty for using long pathway that have inefficient enzymes. So in order to incorporate that, we need to have a description of kind of enzyme usage in each of these reactions. And we do that by incorporating usage of the enzyme in each of the reaction, and the stoichiometric coefficient for that is 1 over the KCat. So you can see if you have a very high KCat, you use very little of this enzyme to carry out a certain flux. And we can then use that to constrain the models, and we refer to this as Gecko. So in order to implement this, we had to collect the KCats. We screened the literature, but also acquired many of them from various databases. In order to account for enzyme usage, so if you have a reversible reaction, we actually now need to formulate both the forward and the backward reactions. So we have to revise the model somewhat. And so we end up with a very large number of reactions and metabolites as specified down there. If we look at the distribution of KCat and molecular weights now in yeast metabolism, you can see here that there is a dominance of about 50 kilodalton, which I guess is a typical enzyme size. Whereas if you look at the KCat, there is a much larger span in the KCat values. And this is kind of interesting that there is such a large variation that is found even in certain pathways. Not like one pathway has very high KCats or low KCats. It's often such that there's a distribution. And that's, of course, because certain chemistries are also just more hard to make. So even though the enzyme may actually have evolved over billions of years, it is still having a relatively low KCat. And so therefore you need to have a high amount of that particular enzyme, as was illustrated also for glycolysis before, that is dominated by a few enzymes, and these enzymes have relatively low KCat values. So could we, with this modeling concept here, begin to describe this Warburg effect I referred to, the overflow metabolism towards lactic acid, which in yeast is often called the Crabtree effect, where it's overflow metabolism resulting in ethanol production. And it is a little bit paradoxical why a cell would do that, because you gain much more ATP when you go full respiration, as illustrated here. But of course, you also need to invest more in building all the proteins or all the enzymes involved in the TCA cycle and the respiratory machinery here. Whereas in fermentation, you just need, in human cells, one enzyme, lactate dehydrogenase, going from pyruvate to lactate, or in the case of yeast, two enzymes to go to ethanol via acetaldehyde. We estimated that in order to generate 100 ATP, we need about 300 grams of protein via this route here, whereas you need 400 grams of protein over here. So you can already see that in terms of getting ATP per protein content, the fermentation is actually cheaper. So what does a cell do? So the cell, when it's grown at limited carbon conditions, restricted growth, and this we can do in using a so-called chemostat culture, where we keep the carbon low and the cells are really energy starved, then they have full respiratory metabolism. We see here CO2 and oxygen production, oxygen consumption and CO2 production is almost following each other. We have complete respiration, RQ of 1. But then when we start to get to higher carbon level, the cell begins to shift its priorities. And in order for the cell to grow faster, it actually has to begin to allocate more for ribosomal system, amino acid biosynthesis, and so on. And now it can save on proteins allocated to respiration, and because as I showed you before, you actually can get more ATP per gram protein by fermentation. And so it starts to produce ethanol here, and it shifts, and that results in a rapid increase in CO2 production and a decrease in oxygen consumption. And the lines are model simulation overlaid on experimental data using this modeling concept, and you can see that the model can completely capture this shift in priorities in cell metabolism. The model can also describe, for example, what are the consequences if we delete certain enzymes in metabolism. In this case here, an NADH oxidase was deleted. This was done in a study from the group of Jack Prunk. And you can see, again, with the experimental data indicated here, the model can correctly shift this, describe this shift in CO2 production or onset of fermentation that happens now at a lower carbon uptake rate compared with the wild type strain, because there's now even less possibilities for doing the respiration. With this multiple and introduction of kinetic constraints, we can also describe much better the growth on a number of different carbon sources as illustrated here. Here's experimental values versus predicted compared with standard FBA modeling that doesn't account for any protein allocation. So we're now expanding this concept to actually describe also all the reactions associated with protein synthesis. So we have all the individual steps of describing the enzymatic processes with transcription, translation, but also protein allocation into the different compartments in the cell, translocation into the mitochondria or various other compartments. And in this, we are having about an additional about 900 genes associated with this. So you can see that we have about 900 genes allocated to metabolic enzymes, but we have about 900 genes allocated for synthesizing these enzymes. So now we're expanding the footprint on the coverage of the yeast genome to about 30% with this kind of model. And the objective, of course, would be that eventually we can design cell factories just like in any other engineering industry where computers are used. But of course, we are still having a lot of dark spots in biology, and so we hope, of course, in this process here also to learn some about in these processes here, and then eventually incorporate this into the models. So then let me talk very briefly about another approach of using systems biology, namely for dissecting the driving differences between different phenotypes. And this is, here we can actually forcefully establish different phenotypes, and it's very well known in industrial biotechnology that you may have strains with different performances. This could be different penicillin strains that have been derived. And it's interesting then to analyze these in order to learn why is this strain better than this one here, and then confer this information to another strain here in order to improve its performance further. And in order to get strains of different phenotypes, the concept of adaptive laboratory evolution has been quite powerful. With this approach here, we expose cells to varying environment. It can be, for example, higher temperatures or low pH or high salt stress or others. And then we select for strains or clones that grow faster at these conditions. And the idea is that they will eventually take over and basically evolve to have improved phenotypic conditions and or properties. And then we can do systems biology to characterize these strains and hopefully find causal mutations and then learn something new about biology. So we often talk about that we can expand this design, build, test cycle also with adaptive laboratory evolution. So we were interested to use this concept to look into high temperature adaptation. So could we get yeast to grow at higher temperatures than it does normally? When we go much above 35, yeast stops growing. And particularly when we go to 40, it goes very, very slow. And above that, it starts to collapse. All eukaryote cell has evolved a system to sustain a response to high temperature at short period of time. This is the heat shock response that onsets synthesis of a number of chaperones in order to protect protein folding and so on. But this is not an inherited response. This is not something that can sustain and allow the cells to grow faster for a long time. So what we did was we therefore did the adaptive laboratory evolution. We then picked up a number of clones that grows at higher temperatures. This is shown here. Here's the blue bar, the wild type at 40 degrees. And here are some of the clones that grow faster at this higher temperature. We then performed genome sequencing of these strains. we found, and we sequenced several of them, we found that some of them had duplication on chromosome 3. There's many reports on chromosome 3 being involved in adaptation to higher temperatures. But we could see that 2 of the clones have no duplication in this chromosome here. We saw that all the clones have mutations in either ERK3 or either ATP3 or ATP2. This is basically forming the same complex. This is the F1, F0 ATP synthase. And so we zoomed in on these 2 and reconstructed some of these point mutations here. And if we introduce point mutations in ATP3, it doesn't confer any improved growth. But if we do it in ERK3, which is involved in ergosterol biosynthesis, we can see that we can recover most of the growth. And interesting what happens when we do further analysis of the biosynthesis, but also on the sterol composition, this mutation here results in replacement of ergosterol to this bended sterol here, ficosterol. And that gives a different folding and different properties of the membrane, so it can actually sustain the higher temperature. This was interesting in the context also because the evolved strain actually have a trade-off. They have lost the ability to respire, whereas this single point mutation inserted in ERK3 recovers the respiration features, and actually there's very little trade-off in terms of growth. There is a little bit slower growth at low temperatures at 30 degrees, but it's not really a serious penalty. We were also looking into tolerance to low pH through adaptive laboratory evolution. This was a little bit more complicated. We did both to an inorganic acid, but also an organic acid, but also only at respiratory conditions. In all cases, we could pick up again clones that have improved phenotypes. We did sequencing, but we also did transcription analysis. Interesting enough, again, we find here aspects, both mutations in ERK5 in this case, not ERK3 as before, but it's just downstream of ERK3 in sterol biosynthesis. We find also different upregulation of the sterol biosynthetic pathway. When we look into the sterol composition, again, what is happening here in adaptation to low pH is a reprogramming on the sterol composition. In this case, you get another sterol compound as illustrated here. However, it is not only this at low pH, and this is also the more typical scenario, that the cell will find several different mechanisms to cope with the imposed stress. In this case here, there's also changed disruption in the iron uptake, and iron becomes more bioavailable at low pH, and disruption of this gene here that is associated with that impacts this. And this can be seen here. Here's one of the evolved strain. Here's when we recover the ERK5 mutations or the iron mutation, and you can see none of them recover completely the phenotype here, but both of them contributed to this. Then it's interesting that if we have an organic acid stress, it's a completely different adaptation the cells are finding. What we are finding that a key dominant driver for mutations here is associated with this AC2 that is resulting in a clumping phenotype of the strains, and so they basically cluster together. But there are also other factors, and you can see down here that deletion of AC2 actually does recover, but not to full extent. We also found a transporter, but that cannot really describe any of the phenotype. So for the organic acid, lactic acid, it's much more complex. We can see that for RNA-seq analysis, that it impacts a number of different things, iron transport, but also a number of different metabolic processes. And it's really, when we compare with the 3 different conditions here, what is driving these differences here, we can see that proton symport, not surprisingly because we are at lower pH, becomes a real key driver, and regulation of this has to be downregulated in order for the cells to survive. And so you are in a situation where you are imposing stress, and you can have 2 different paths. One is on adaptation to an inorganic acid, and one is to an organic acid, and you find completely different routes for this. And that's kind of interesting also, of course, in the terms of evolution, but also how we can use systems biology to quantify and find causal mutations. So then let me move on and talk a little bit about cancer metabolism. So metabolism of cancer is rewired to balance proliferation and survival. There's been a number of studies reporting on this recently, and metabolism of cancer is probably one of the hottest fields in cancer biology today, and also because there's developing several drug targets that are enzymes that are in the phase of being evaluated for attenuating or altering the metabolism of cancer cells and hereby killing them. So we have therefore been working on developing human genome scale metabolic models. We call our model HMR2. It's quite comprehensive. And again, with the objective is that we can develop we have a generic model. We can also develop specific models for different tissues, but also different cancer types. We can compare the networks, for example, between cancer and a normal cell. The model we've generated is HMR2. It's generic. We then have this init or t-init algorithm that can be used for generating different cell lines or human cell types. And we have also manually curated it for hepatocytes, adipocytes, and myocytes. So one example of cancer was that we were interested to look into the heterogeneity of cancer metabolism. So we took, in this case, that was proteomics data based on immunohistochemistry for HCC, liver cancer, the most dominant form of liver cancer. And here's for standard hepatocytes staining. Here are the different proteins. And then for the different 6 different biopsies from 6 different patients. So we have these liver biopsies, we have the antibody staining, so we then ran this init algorithm to find models for each subject, basically, for each cancer type. And there were quite some variations, as you can see. This is just mapping on the enzymes. You can see that this patient here, in that cancer, in that tumor, all these enzymes are basically lacking, whereas this one over there has a special phenotype, this one also has. So there are quite some large variations between the different tumors. We then wanted to see if we could find computational targets that would hit the metabolism in all 6 subjects or all 6 cancers. But then we also, of course, wanted to have that it would targets that would not kill metabolism in the healthy cells. So we ran a search for so-called anti-metabolites that will kill the tumor or prevent tumor growth in the 6 tumors, but not prevent growth of the 83 healthy cell type models. So this was kind of a toxicity test. And then we found a number of targets, and several of them were consistent with earlier reported targets for cancer treatment. But we found also a new target, which is involved in acyl transport, acyl fatty acid transport into the mitochondria for beta oxidation. So this is a carnitine carrier system here. And if that is blocked, the cancer cells cannot use fatty acid as an energy source, and that significantly reduces their ability to grow. And so therefore we wanted to test that marker. So we took a cancer cell line for HCC, HepG2, and we benchmarked it with sorafenib, which is used for treatment of HCC today. And we could see that, particularly when we went up in concentration here, that the perhexylene could actually then attenuate growth of this HepG2 cell line, indicating that this is really an important factor for driving energy generation in these cancer cell lines. So we were also interested to look into, is metabolism of cancer cells different from normal cells? So for that study, we took data from the Cancer Genome Atlas for 10 different cancers. The nice thing about these data here is that they are data on RNA-seq from both the tumor as well as from the healthy tissue from the same patient. And so therefore we could begin to look at what is the difference between the healthy tissues and the tumor tissue. And one thing that was sticking out completely when we did this was that one cancer type, clear cell renal carcinoma, was completely separating, as well as also the metabolism of the healthy kidney here. So we looked further into that and just looked at the metabolism or the expression from the cancers, and there you can see that clear cell renal carcinoma is separating completely from the 9 other cancers. So why is this so different? Well, this cancer type is relatively unique in the sense that it accumulates quite a lot of fat, and that is also where the name comes from, the clear cell. It does have a number, it has the Warburg effect, as many tumors has, but it has really also enhanced the pentose phosphate pathway with NADPH generation, and then, as I said, this lipid accumulation. So when we did, we reconstructed then genome-scale metabolic models for the healthy kidney as well as for the tumor. And one surprising thing we found was that this cancer type is characterized by a very large loss of function and very small gain of function. So this is seen here. These are number of genes. Only 56 new genes are activated in the tumor compared with healthy healthy kidney, whereas 885 genes are downregulated or completely not expressed anymore. And so this completely changes, and this we could actually also use for studying whether we could find new targets for killing the tumor, because it's now more vulnerable, it has less metabolic flexibility compared with the healthy kidney, and that can be used for drug target. But one thing that we stumbled across that was kind of very interesting was that there was a very remarkable dysregulation of glycosaminoglycan biosynthesis, particularly chondroitin sulfate and heparan sulfate biosynthesis. And this you can see here. This is a map of the network of the biosynthesis of these glycosaminoglycans. Red means upregulated and blue is downregulated. And if we overlay that on the biosynthetic maps of these, you can see here that chondroitin sulfate biosynthesis is significantly upregulated and heparan sulfate is downregulated. So that led us to speculate that we could analyze these glycosaminoglycans in plasma and urine. And so we collected data or samples from first 22 patients and 8 controls. And we measured these compounds and we could see a distinct separation. So we then did further analysis in validation cohorts, and we have now actually done this in a total of 250 subjects. And we have a very, very distinct separation, and we are now carrying this forward as what we call a systems biomarker for diagnosis of kidney cancer, but also as a diagnosis for whether it's a benign tumor or an active tumor observed. So let me end talking very briefly about modeling of the human gut microbiome. So I guess you're all aware that we live in this microbial world. We have a lot of bacteria harboring the human gut. We have about 2 kilos of bacteria in the human gut. They play a very important function in determining and impacting a number of different diseases. We can analyze these by performing metagenome sequence analysis. This gives a lot of data, and we are interested in translating that into a specific metabolic map. So in one study, we were looking into if there is a correlation between the gut microbiome composition and symptomatic atherosclerosis. It was a relatively small study, 12 patients and 15 controls. We had kind of extensive metadata. What was interesting was that just a PCA analysis of the metagenome data allowed for a separation between control and patients. There were two controls here that were here. It turned out that they had a minor atherosclerotic event later on after the samples had been sampled, and when they were removed, we had a very clear and distinct separation. And interesting enough, we could find also what really drives this. There are some specific species that are driving this, but more importantly, when we look into what is there any metabolic drivers of this, that it turns out that the genes associated with beta-carotene and lycopene biosynthesis are really distinctly different between the two groups. And that led us to begin to analyze for beta-carotene and lycopene, and you can see particularly beta-carotene, we had a significant separation. It's much more elevated in the control patients compared with the patients with disease. We also looked into the impact, whether we can analyze type 2 diabetes. This was a larger cohort study, about 150 people, type 2 diabetics, impaired glucose tolerance and normal glucose tolerance. And if we just look at the type 2 diabetics versus the healthy controls, we could find correlation between identified species and a number of plasma markers and other markers. And you can see overall we have over here what we call a bad profile, a set of species that seems to be more abundant in subjects with type 2 diabetes and thereby alter the gut microbiome metabolism in a bad direction. So here it of course becomes interesting if we can begin to model this behavior, because then we can also predict which probiotic should we give to these patients in order to steer the gut microbiota in a certain new direction. And so that's why we are turning and taking data from human observation, doing modeling, then doing evaluation through our collaborators in Gnobiotics mice. And so here, I'll just end with one slide where illustration of how we did that. In one study, we took abundance data for two different groups of subjects and converted and then developed models for the 5 most abundant species. And with those models, we could actually correctly describe the changes that happens in amino acid in plasma when these two groups of subjects are undergoing dietary restriction. And so this is a baseline. This is after dietary restriction. You can see that it's moved over here. And the models, I'm not going to go into details here, could actually correctly predict these changes that we observe. So with that, I'm not going to give any conclusion here. I'll be happy to take questions. Let me acknowledge all the people who have been involved with this work here, the funders, and you for your attention. Thank you.